Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for UW Extension Cooperative Extension, and on behalf of those folks and our other co-organizers, Wisconsin Public Television, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight it's my pleasure to introduce to you Kate O'Connor-Giles. She's a professor here in the genetics department and the cell and molecular biology program. She was born in Champaign, Illinois. She grew up in Champaign, Illinois and graduated from Champaign High School, at which time she went to the University of Illinois at Urbana-Champaign, where I spent 2 of the most glorious years of my life. Then she went to Washington University in St. Louis and got her PhD. She came here to do a postdoc with Professor Barry Genetsky, and now she's on the faculty here. She gets to talk to us tonight about CRISPR, which is probably the single most interesting new technique as far as breadth and how it's shaking up the wide fields of life science since polymerase chain reaction. At least that's my take on it. It's going to be very interesting, as you can see, with the eraser. A very interesting way to edit genes, maybe one base at a time, maybe one gene at a time. So please join me in welcoming Kate O'Connor-Giles to Wednesday Night at the Lab. Thank you very much.
Okay, thank you very much. Can you hear me? Okay, so just before we start, how many of you have heard of CRISPR in the media? Okay, following it closely? A few of you. Okay, great. Great. Well, so as many of you know, it has been a longstanding goal of biologists to be able to do exactly this, to erase and rewrite genomic sequence, okay? And so this has made, this new CRISPR technique has made a big splash. It was the breakthrough of the year in 2015, according to Science magazine. Nature just last month came out with an issue dedicated to CRISPR called CRISPR Everywhere. There was an interesting article called The CRISPR Zoo that you might want to read if the topic is of interest to you, of particular interest to you. It really talks a lot about the many, many things that we can potentially use this new genome engineering technology for. And before I go into the details of how it works, I thought that I would just highlight some of the things that it has the potential to affect, and in many cases is actually already affecting. And so those things include model organism research. So those are actually mutant flies that we generated using the CRISPR technique. I'll tell you a little bit more about that later. Correcting disease-causing mutations. So if we can go in and change a disease allele to a wild-type allele, that would be obviously something that would be of great potential value for human well-being. Food production. So we all know about GMOs. This is a way to modify the genomes of both plants and livestock in a way that doesn't involve bringing in foreign DNA. Okay, so this may be a potential way to improve our ability to feed the world. De-extinction, which you guys are going to hear about soon. So people are actually very serious about using CRISPR technology for de-extinction. So you'll hear some about, you know, the maybe the more Jurassic Park type cases, like bringing back the woolly mammoth. One of the big leaders in CRISPR is actually extremely interested in this prospect. But there are other things, for example, heirloom tomatoes. Other varieties of foods that are no longer available to us that we might be able to take modern varieties, edit them to make them match varieties that are no longer available. Biofuels, and really many, many other things. Pets. This is happening, not in the US, but editing animals, There's a Hercules dog that has large muscle mass that was made using CRISPR. And then targeting disease vectors. And that one I'm actually going to focus on at the end because this one is really of just dramatically increasing interest with the Zika virus. And I think that's the coincidence of this technology and that the emergence of this mosquito-borne disease actually has a real potential to change the course of what might happen. Okay, so what is CRISPR? So I'm going to use typing a Word document as an analogy for what we're doing with genome engineering. And so basically the first step that is finding the spot in the genome that you want to edit. Okay, so you need some mechanism for searching. So here what I've done is I've typed germline correctly twice and incorrectly once. So I'm trying to find the one time that I typed it incorrectly, and I failed. I found it all 3 times by typing in germline. So I had to revise my search and add the period, and then I found the correct spot. So I needed precise localization to the spot that I want to edit, and then I need a way to get my cursor in. So I need to break into that spot so that I can then go in. And here I'm just showing this magnified. So I've got my cursor in the right spot. I've broken into that word, and now I can type in the E and correct this. And this is actually really quite analogous to what we're doing with genome engineering. So DNA breaks initiate genome engineering. They initiate genome engineering because they initiate DNA repair. And what genome engineers are doing is they are trying to essentially hijack the cell's normal repair pathways. And in order to do that, you first have to trigger them, okay? And so you've got a double-stranded DNA break. That initiates 2 main pathways. There are some variations on these themes, but there are 2 main ways that a cell can repair DNA. And so this is obviously a really important job for a cell, right? So things like UV light can break DNA, X-rays, and cells have evolved to handle this. And so one way that they do it is simply through just ligating the 2 broken ends back together. Okay, this is called end joining or non-homologous end joining, and it is exactly what it sounds like. It's a ligation of the 2 broken ends. Now the problem with this is that this is an imperfect process, and you can actually lose a base pair or several base pairs or gain a few at this junction. Okay, so it's an imprecise process. The other pathway is homology directed, and what that means is that the broken end goes out and looks in the genome. It's looking for the other allele. Right? So there's a perfect copy somewhere else in the genome if you've broken one chromosome. It's going and looking for that, and then it uses, once it finds it, it uses that as a template to synthesize across the break and precisely restore the original DNA. Okay? So any questions about that? Okay, great. So these are the 2 pathways that the cell has available to it. And so for a long time, the really big challenge was how do you go in and make a precise break at one spot in a genome as huge as the human genome or even as big as the fruit fly genome? How do you do that? And there were ways to do that. So I guess I should back up a little bit and say the first thing you need to know sequence. So you need genome sequences. We've got that, as you're all probably well aware. We have many, many, many organisms' genomes sequenced. So you need the genome sequence, but you need more. You need a molecule that you can target to that sequence. Okay? And so there were proteins that did this job. These proteins have actually been around for a couple decades. So CRISPR is not something that's completely new in the sense that it allows us to do something we couldn't do before. What is new is that it allows us to do something, it makes it feasible. It makes it reasonable to do it in a timely and an efficient manner. These proteins were very difficult to make. They didn't target perfectly, and so they were not really used by many people, even in a research setting, let alone in some of those other settings that we were talking about. And so many people, us included, were working very hard to try to find better ways to make a targeted break in DNA. And I'm not going to talk about this very much, but the couple things that our lab was working on, we were building on the work of Bill Engels and colleagues from the 70s and 80s using transposons, so the jumping genes discovered by Barbara McClintock in corn. When they jump, they actually break DNA. And so we were working on using those as a tool for making targeted breaks in DNA, because in many model organisms, we have large collections of lines that have these transposable elements all over the place. We were also working in collaboration with Mike Cox and Jill Wildonger from the biochemistry department to try to bring a system from phage, which are viruses that infect bacteria, into other organisms. In our case, we were trying to bring this into fruit flies because that's the organism we work with and the organism we desperately wanted to be able to just erase and change base pairs in. And so we were working on this, and this is just sort of to give you an idea of kind of the setting. We were, three of many, many, many people around the world who were trying to solve this problem when these papers came out in January of 2013, and it became instantly clear that the problem was solved. And so these papers showed the use of the CRISPR system to make targeted DNA breaks. And it was immediately obvious to all of us who were working in the field that there was not going to be an easier way to do this, that this was so programmable that it was going to be difficult to top. It could be improved, but the basic concept was what everyone had been dreaming of. And so I'm just going to kind of walk through quickly how it works. So basically what happens is you pick a 20 base pair target in the genome. Okay, so pretty much any 20 base pairs to target. You make an RNA that is complementary to that target. So it can bind that target through base pairing, through simple base pairing. So the logic is easy. The molecular biology is extremely easy. This is will cost you, $20 and take you a day, and really a couple hours worth of work over that time. So this is really easy molecular biology for any lab. Easy to program. Okay, so what that RNA does then is it binds this protein called Cas9, which is CRISPR-associated 9. Cas9 then is brought to that specific DNA target with the guide RNA. Cas9 is a nuclease, which is a protein that cuts DNA, okay, cleaves DNA. It has 2 nuclease domains, so it will cleave each strand of the DNA. And so that's it. It's as simple as that. You make an RNA that is complementary to the sequence in the genome that you want to target. You express this protein, Cas9. It will come along for the ride with the RNA to that spot in the genome and make a double-stranded break. Okay? So I think you can see, why we were all so excited. This was amazing. And so I just kind of want to walk through the history of this. I think that some of the popular literature, or in particularly in the media, sort of suggests it came out of the blue. And as all of you follow science, you know that's not how science works. And so we're going to start our story in 1987. So what happened in 1987 was a group was just cloning some gene in bacteria. They were just working on their gene of interest. And they noticed next to their gene, when they sequenced it, that there was this weird sequence. It had these weird repeats, and then with sequence in between that didn't match anything that they knew. This was before any genomes were sequenced, so that it didn't match anything wasn't particularly surprising. But it was this real weird, regularly repeating sequence. They mentioned it in their paper, oh, by the way, downstream of our gene is this weird sequence, we don't know what it is. Over the years, by say 2000, there had been about 30 bacterial genomes sequenced, many of them here at Wisconsin. And they started to recognize that these sequences were actually common in bacteria. And it turns out that 40% of bacteria have these CRISPR sequences, which stands for clustered, regularly interspersed, palindromic spacer something, sorry. Short palindromic repeats. I always get that part wrong. So it's something that I don't even remember and never say, so you don't have to know it. It's basically clusters of repeated sequence is what CRISPR means, okay? Okay, so by 2002, people who work on bacteria and were interested in bacterial biology were working on this and interested in this really weird sequence. It must be something interesting. It's conserved. They found that also conserved were these CRISPR-associated proteins. So we talked about Cas9, so CRISPR-associated 9. A number of proteins were found to be always adjacent to these CRISPR repeats, and they encoded enzymes that manipulate DNA. So enzymes like nucleases that we just talked about that cut DNA, enzymes that unwind DNA, things like that. So this was starting to give them some hints. In 2005, multiple groups reported that those sequences in between the regularly repeating sequences were invaders, like viruses. So they were sequences from viruses and other things that invade bacteria that they may want to defend against. And so in 2006, Eugene Koonen's group hypothesized that this was actually an adaptive immune system. So this was something that certainly was thought to be specific to mammals, that you could actually encounter a pathogen, have some way of remembering it, and going back and fighting it rapidly the next time you encountered it. And this was tested in 2007 in Madison. So this was a group working at Danisco, which is a yogurt company in Madison. And so cheese plays a huge role in the importance of this. So they made yogurt and pizza cheese, and their cultures kept getting contaminated. So they were very, very, very interested in, for their yogurt cultures, finding ways of protecting them from these viral infections. And so they tested this hypothesis, and what they did was they just selected bacteria that could withstand viral infections, the ones that survived, sequenced them, found out they had CRISPR sequences from those viruses. Then they challenged them with another virus, sequenced the ones that survived, and they had incorporated into their genomes sequences from those viruses. And so they were able to prove, and then also they were able to show that then the next time they were exposed to those viruses that they were again immune to them. And so basically over that 20 years, microbiologists were able to figure out that what happens is that when a virus invades, let's see if I can do this with my pointer. Okay, a virus invades, the bacteria actually snips out a piece of its DNA, sticks it in between these regularly repeating sequences in its genome. When it encounters that same virus again, it actually transcribes those sequences, makes that guide RNA that then is complementary to the virus DNA. It leads a nuclease or a complex of proteins, including a nuclease, to the viral DNA and cuts it up, killing the virus. So, pretty cool. So, why did it take 5 more years for people to think about using this to solve that problem that I just presented at the beginning of this talk. It wasn't that it took them so long to think about it. It was that there are many different types of CRISPR systems in bacteria, and it took that long before one that was simple enough, that only required one protein and 1 or 2 RNAs was found. The others required large complexes of proteins that would have been prohibitive for getting those into other types of cells. And so these 2 groups, Jennifer Doudna and Emmanuelle Charpentier, and then the Sickness group, again with Barangu, who was one of the guys who worked in Madison at the yogurt company and was one of the first to figure out its function in bacteria. What they showed was that you could use this in a test tube. So in a test tube, if you put in the protein and the guiding RNA, the Cas9 protein and the guiding RNA, you could cut DNA at will. So you could program it to cut DNA at specific sequences. So this was really exciting. And I just want to tell you how their system works exactly. So I have to add one component to the story that I told you, which is this thing called the PAM, which is a protospacer adjacent motif. And what that is, it's very important because if you think about it, I just told you that the bacteria insert the viral sequences into their own DNA, right? They have to have some way of telling self from invader, and they can't go around cutting up their own DNA. This is it. Every bacterial species will cut next to a specific sequence. So when it snips out a piece of sequence, it only snips out sequences adjacent to a specific sequence. In the case of Streptococcus pyogenes, which is the one that most genome engineers use, that sequence is NGG. So it's not particularly limiting, but basically it means that as genome engineers, we have to target sequences. We can target any 20 base pair sequence in the genome as long as it's next to an NGG. If it's not next to an NGG, the CRISPR-Cas9 system won't cut it. Okay, so that's a reason why people are trying to gather these systems from many bacteria that have different PAM sequences so that we can cut everywhere in the genome. And so it's not going to be a limitation. There are gazillions of these that have evolved. Okay, and so I'll just point out also that this guide RNA that we use is actually a chimeric RNA that was made by those 2 groups and discovered to work. They're in the endogenous system in the bacteria. It's actually multiple RNAs. But so it's been simplified to this system that I described earlier. And so using this approach that had worked in test tubes, 3 groups in January and February of 2013. So they showed that it worked in test tubes in August of 2012. By January of 2013, these groups had shown that it worked in human cell lines. Okay? So the potential was immediately recognized. Within another month or so, other groups had gotten it to work in zebrafish and in yeast, so some key model organisms. We, in collaboration, so this is Scott Gratz, a graduate student in my lab, and our collaborators, Melissa Harrison from biomolecular chemistry and Jill Wildonger from biochemistry, we got it to work in Drosophila by May of 2013, and were able to demonstrate for the first time that this system could actually be used to edit the germline and to transmit changes to edited DNA to the next generation. And so this kind of gives you an idea of the fact that it is actually pretty easy to get working. And pretty much every organism in which people have tried to get this to work, it works. So these are just a sampling of some of the many organisms in which CRISPR genome engineering has been successful. So your model organisms, mice, followed shortly after flies, Arabidopsis and other plants, pigs. There's a big initiative to model some human diseases in pigs, which make better physiological models than mice in many cases. Monkeys, butterflies, which many of you may know are studied in evolution and development. And then mushrooms just came out. How many of you saw the article about the CRISPR mushrooms? Yeah, so just last week, I think it was, or 2 weeks ago, the USDA passed on regulating CRISPR-engineered mushrooms. So these mushrooms were engineered using CRISPR genome engineering to knock out the function of a gene that's involved in browning. And so what this does is increases the shelf life, decreases the browning of the mushrooms. And because the change involves no foreign DNA, the USDA said it's not a GMO. And so that was pretty big news. Okay. All right, and so I just wanted to also, I imagine that many of you who are following this are also reading about the patent wars. So there are some major patent wars going on, and luckily I work in fruit flies, so nobody cares, there's no money to be made. So I just wanted to give a shout out for the other side of this. So the clinical applications certainly have, and agricultural and many others have great potential to make a lot of money, and so this patent is important. But the groups involved, so Feng Zhang's group at MIT, and many other groups have been extremely open and generous in sharing with researchers. And that's a major reason why this technology has spread so far and wide in labs. And so we, for example, have a website and some tools to help that helped people get going in fruit flies. There's similar things in worms. And so I just kind of wanted to show the other side of this, that it actually has been a remarkably open process that's evolved incredibly rapidly with lots of people sharing unpublished information to help pass on this really important lab technology for nonprofit research labs. Okay, so I told you that this solved that problem, the problem of how do you go in and make a break at a specific place in a complex genome. That's not to say that there aren't ongoing challenges. This system can and needs to be improved. So we need to increase the specificity. People are working on this. This is happening. 20 base pairs isn't that many, and it actually can bind and cleave at not just the identical sequence, of course, if that occurs more than once in the genome, but at similar sequences in the genome. So those are off-target effects. We also need to increase the efficiency. So the efficiency is much, much higher than anything in the past, but it's nowhere near 100%. And then here's the big one. We can make the break, and then we pretty much can cross our fingers and try to bias things, but we don't have control over which pathway the cell chooses to repair that break. Okay? So all we can do is try to bias it. And generally speaking, that means biasing it against end joining, which in many cells is the preferred pathway, towards homology-directed repair, where the cell will repair not off of the homolog or a sister chromatid, depending on the point in the cell cycle, but off of an experimenter-provided piece of DNA. Okay, so to make the edit that we want to happen. That is a huge challenge, and it's not going to be solved anytime soon. So I think the first one is easier, and that one we're going to get to faster. So I think that we don't have to worry, for example, about designer babies next year, or perhaps in any of our lifetimes. On top of the fact that we wouldn't even know what DNA to change, we can't change the DNA the way that we want to. We do not have that kind of control. So why then do we see? Well, so let me actually back up a little. I wanted to give another analogy for that path, the not being able to control the pathway. And this time I'm switching to text editing. So how many of you text? Not a lot. Okay, half of you. So it can be incredibly frustrating because your phone is competing with you to fix your mistakes. So I, being a Drosophila researcher who works on CRISPR, often, will send texts along these lines that I'm sure my students are thrilled to get. CRISPR can be used to edit the Drosophila germline. So I left out the E. I again need to get my cursor in and go correct it to add an E. But my iPhone thinks I'm trying to say gremlin. And so I have sent many texts about Drosophila gremlins because my phone wins and I lose. And so that's essentially what we're dealing with in the cell. The cell may be trying to write gremlin, and that's not right. That's not going to help the disease state. So it's a real challenge. Okay, so why? Why then are we seeing headlines like this? The CRISPR craze, jaw-dropping breakthrough, could change biotech forever. First of all, there's the prospect. But also, there is the fact that under conditions where we can select for the events that occurred the way we wanted them to occur, this is paradigm shifting. This is changing the way we do things. This is a drawing that one of the undergrads in the lab did for us. And this is really how we think of CRISPR. We can make any fly we want to make now. We've got a little machine, we put in a wild type fly, and out comes the fly we want. It's fabulous. We really have high efficiency, high success rate, but we can toss the flies that aren't what we want. So that's a very, very, very different situation than a clinical setting, of course. So the same is true for seeds, right, and things like that, where you could select for the change that's the change that you want, toss anything that has an undesired change. Okay, so I thought I would briefly tell you, what we're using this for in my lab. So we're interested in a big question: how do you build a brain? And more specifically, we're really interested in understanding how you build a functional nervous system that can be both reliable on the one hand, so reliably control behavior, but also plastic, change in response to the environment. And so I have a little video here that I hope is going to play. Oh, I'm afraid it's not. Oh, it's such a bummer. I'm going to describe it because it's so cute. It won't be as cute without seeing her. So this is a kid who has an iPad. And so she's learned how to use her iPad with her fingers. And then she goes to use a magazine, and the magazine is broken, and she gets really frustrated, and she checks to see if it's her finger that's broken or what's going wrong. But I really love it because it's an example of overlearning, right? And so your brain needs to be reliable, but it also needs to be modifiable. And so this is a really complex problem. The behavior, of course, is controlled by the brain. The brain is organized into neural circuits. These neural circuits, their function is dependent upon the component neurons and their interconnections and their communication at synapses. And so my lab actually focuses on synapses. And so we're really interested in synapses, both because these are the fundamental unit of communication in the brain, but also because they're the site of plasticity. So this is where the changes in brain function that underlie learning and memory occur. They occur at synaptic connections. It's also becoming increasingly clear that aberrant synaptic function underlies many diseases, many disease states. And so we think that in order to understand the brain, you need to understand synapses. And of course, I'm a geneticist, and underlying synapses are genes, right? And the genes that code for proteins that function at synapses. And so what we're doing with CRISPR, and the reason we were so interested in being able to edit DNA, is we're trying to take advantage of the genomic information that's increasingly available to identify candidate new genes that might be involved in synaptic function that are conserved from fruit flies all the way to humans. And then what we want to do, once we identify good candidates, is knock them out. And so we use a CRISPR-based technique to essentially just slice out the gene. We actually replace it with a red fluorescent protein that makes their eyes red fluorescent, the fruit fly's eyes, because that allows us to follow the mutation, and it's fun. And then what we do is we then screen these. So we've made mutants in these genes we think are important, and we test whether or not they have altered synapse morphology, altered synapse function, and ultimately altered behavior. And in this way, we're identifying interesting new genes. And then once we find new genes, we can use CRISPR to actually really get at their function. So we can start to make more informative mutations than just getting rid of the whole gene. We can change specific amino acids. We can knock out specific domains and really, probe their function with mutations. We can also, and this is one thing we commonly do, stick in a tag. And so how many of you have heard of GFP, green fluorescent protein? Maybe seen green fluorescent worms that you can buy at the grocery store. So this is a jellyfish protein that biologists use a lot. And what we can now do is we can stick it into the genome and hook it up to our genes of interest so that when the cell makes our protein of interest, it will make a fluorescent version of that protein. And we can follow it in cells. We can follow it in behaving animals. And so we can see what these proteins are doing at synapses under different scenarios. And so CRISPR really is an incredibly valuable tool to us in the lab and has changed not just the way we do things, but the things we can think about doing. And so with the time remaining, though, I wanted to talk more about some of these other functions. Okay? So specifically, I want to focus on the idea of correcting disease-causing mutations in humans and targeting disease vectors. So I think these are some really important potential applications of CRISPR technology. And so how many of you read about the CRISPR editing of a human embryo? Okay, very few. So, shocker. So this has been done. This was done in China. Human embryos have been edited using CRISPR technology. The embryos were non-viable. So they were triploid. They had been, so it was, in IVF sometimes the egg will be fertilized by two sperm. And so that's not compatible with life, and that's the embryos that they used to address potential ethical concerns. And so what they were trying to do was edit the gene that's mutated in beta thalassemia, an inherited blood disorder. And so I just wanted to kind of walk through what happened. So they injected 86 embryos. Of those, 71 survived 48 hours. So, so putting the CRISPR-Cas9 components, the guide RNA and the protein in, wasn't too toxic. They sequenced 54 of those. Of those 54, half were edited. So half, there was no evidence of anything having happened. Okay? I'll point out that we can't tell the difference between nothing happening and the cell correcting it properly. Okay? So, correcting it right back to what it was before it was cut. Okay, so of those 28, 17 had those indels, which are the insertions or deletions that I talked about that can occur when you use the end joining pathway, this sloppy, error-prone, I should say, pathway. And so they were potentially worse than they had started out. 7 used the homology-directed repair pathway, but instead of repairing off of the corrected template, DNA template that was provided by the experimenters, repaired off of a very similar gene in the genome. Okay? And so you ended up with the wrong gene. And then in 4 cases, repair did occur with the donor template. So we're at 4 out of 86. Okay, so not great odds for a clinical application. And I should say, so some ethicists have come out and said that, that this is ridiculous, this is just playing with human embryos. Everything that they saw, we see in the lab, everyone has seen in every organism they work in. This is not new information, this is not unexpected, this is not a surprise that this is what happened. This is what happens when you do CRISPR. Other things that happen when you do CRISPR is off-target cleavage. So I told you that the guide RNA can bind similar but not identical sequences. That happened in this case. So even in those 4 where they got the repair that they wanted at the site they were targeting, they had off-target cleavage. They also observed a lot of mosaicism. So the 2 alleles would repair differently in a cell. Different cells would repair differently. So they didn't get whole organisms where everything had happened properly in all of the cells. Okay, so again, designer babies are not imminent. But this certainly does have people thinking more about the prospect. And so I just wanted to mention that there are lots of things happening to try to think about the ethics of using CRISPR, Cas9, or any other genome editing technology for that matter, to edit human embryos or humans. So it doesn't necessarily have to be embryos. It could be somatic cells in a mature human. And so these are just some of the initiatives, and I wanted to highlight that our very own Alta Charo, who is a bioethicist in the UW Law School, is very actively involved in this. And they held a town hall a few weeks ago that was really interesting on the ethics of CRISPR editing. So you might keep your eye open and see if they do that again. But very informative. Okay, so I'm going to switch topics and try to hit one more application before our time is up, and that's the Zika virus. And so as many of you know, this is a devastating disorder. It's currently affecting people in South and Central America. Pregnant women who are infected can give birth to children with microcephaly. And it is carried by the mosquito Aedes aegypti. So what people want to do is something called a gene drive. Have any of you heard of a gene drive? Okay. I had not heard of a gene drive until about a year ago. It's a very interesting, think of a selfish gene. So it is a way of making sure that one allele is passed on to 100% of progeny. And so this deviating from normal inheritance patterns allows the new allele to spread rapidly through a population and take over. Okay, so how could you do that with CRISPR? So it's kind of diabolical. And so they did this in flies. And what they did was they put in a guide RNA and they put in a donor template to target a gene. But what they did, that donor template actually had the DNA to code for another guide RNA and another Cas9 protein. And so you replace a gene with the molecules that will kill the other copy. Okay? So allele 1, you cut, Cas9 comes and cuts, homology-directed repair occurs, and you incorporate the coding sequence for Cas9 and the guide RNA targeting that gene into where that gene used to be. So homology-directed repair occurs, and that is now replaced the gene. So then what happens to your second allele? Well, that first allele, the Cas9 is expressed, the guide RNA is expressed, and it targets the second allele and converts it to itself. This time, instead of using an experimenter-supplied DNA template for repair, it uses the homolog. And it incorporates the Cas9 and the guide RNA into its gene locus. And so you end up homozygous for this. You pass on one copy to your offspring, they convert the other copy, and they are guaranteed to then pass it on to their offspring. And so it rapidly spreads through the population. This could be something that makes the animals die young. It could make them not mate. It could make them not able to be vectors for a particular virus. It could make them all male. You could do all sorts of things. And so this was done in fruit flies, and it's now just last month been published that this was done in mosquitoes. And so this has raised a lot of concerns about what the potential implications of such a gene drive could be on the environment. And so many of us who work with CRISPR-Cas9 genome engineering, in particular in flying insects, where there's a reasonable risk of escape from a lab, got together and wrote a policy statement. And you'll note that this includes not just those of us who are using CRISPR-Cas9 for other things, but the authors who were creating these gene drives all agreed on safeties that needed to be in place in order to use these technologies that could potentially create a gene drive in the lab. And I'll point out that nearly everything everyone's doing in a lab could not possibly create a gene drive. So that it takes a particular type of experiment to cause that. And so this is what's happening on the lab front, but it'll be interesting to see. I read a survey where the numbers were very, very high in terms of public support for wiping out Aedes aegypti if we can do it. So I don't know what's going to happen. But it'll be interesting to see, and CRISPR certainly does make it more possible. None of my movies are playing. So sad. So this was a worm. This was not a worm. It's a Drosophila larva, and it has calcium indicators at all its synapses, so you can see them light up as it moves. Sorry, I screwed up my movies. But I would like to thank the members of my lab, the current members, past members, and some of the undergraduate researchers who have worked with us on the CRISPR-Cas9 development side of things, as well as our collaborators, Melissa Harrison and Jill Wildonger, and our funding sources. Okay, thank you very much.